Hello, everyone, and a warm welcome back to our Evolution and Ecology seminar series. My name is Elizabeth Duxbury, and I'm a postdoc in the research group of Alexei Maklikov at the University of East Anglia, and I'll be your host for today's session. Myself and our wonderful team of co-organisers, Andreas, Ulia, and Wouter, are very grateful for the enthusiasm from the research community for this initiative, and it has been a pleasure to host such a variety of great minds so far. So thank you all for your fantastic engagement in the series. Before I introduce today's speaker, I'd like to remind you that there will be a Q&A session directly after the talk. So please post your questions in the dedicated Slack channel and upvote questions that you'd like to hear. We aim to keep the whole session to 1 hour in length. So it is my great pleasure to introduce today's speaker, Dr. Rob Salguero-Gómez, Associate Professor of Ecology in the Department of Zoology at the University of Oxford. Rob's research explores the ecological and evolutionary drivers of the remarkable diversity of life history strategies seen across the tree of life. Rob's work combines population demographic modelling and comparative analyses with life history theory and field experiments to address fascinating questions such as why can some species apparently escape from senescence, and what metrics can we use to predict species resilience to environmental challenges, including climate change? Rob is also a strong advocate for open access reproducible research, and he founded the freely accessible Comadre Animal Matrix database of demographic data during his postdoctoral position at the Max Planck Institute for Demographic Research, in addition to later coordinating the Compadre plant database, which together provide a very valuable resource for the research community. In recognition of his contributions to ecology and evolution, Rob was awarded the prestigious British Ecological Society Young Investigator Prize, and also co-authored a highly cited Nature paper on the diversity of aging patterns across taxa. Today, Rob will present his talk entitled Using Insights from Life History Theory to Forecast Global Responses to Climate Change. So Rob, thank you for accepting our invitation, and over to you.
Thank you so much. Can you hear me okay?
Yes.
Fantastic. So thank you so much for the very kind invitation. I think it's very important for the ecological and the biological society at large to try to carry on with the dissemination efforts that we are all committed towards. And on that note, please allow me to commend you and your collaborators for this wonderful seminar series. I was just watching Jess Metcalf's conversation last Friday, and I really enjoyed it. So thank you so much for allowing me to be part of that and to share some of my research. Understanding where species are found now, and very importantly into the future, is one of the most pressing ecological and societal needs that we are faced with. The take-home message of what I would like to point out today is that life history theory and the data that underlie the theory, the demographic data, together form a perfect marriage in order to be able to provide powerful climatic forecasts that would allow us to understand how species, where species, which species might no longer be with us due to climate change, and which species might persist. Some of the research that I've developed during the last few years takes some major inspiration from older disciplines or older sciences like mathematics, physics, and chemistry. One could argue that ecology is but starting to leave its teenage phase, in that we are now verging into the ability to forecast, the ability to predict. And this is something that I think chemistry, for instance, among other older disciplines, has done wonderfully. Take, for instance, perhaps the most well-known item within the curriculum of any chemistry undergraduate, the periodic life table of the elements. The periodic life table of the elements in chemistry organizes elements according to two very simple properties. How many electrons are there in the outermost orbits, and how many orbital layers does the element have? You know those two key pieces of information, and you will be able to place the elements along the table. You will be able to know what that element is likely to bond with other elements to give rise to molecules. You'll also be able to predict between molybdenum and ruthenium there being an element that by 1913 had not yet been discovered, but that in a few years, if you give me a few seconds, will be filled in. So the ability to classify, there you go, technicium. The ability to classify is really important because it allows us to be able to predict as a next step. And this is very much the foundation and the inspiration for the kind of work that we conduct in the Salgo team, my research group at the Department of Zoology, Oxford. So I mentioned before that the take-home message was you marry life history theory with data and you're in a good place to do ecological forecast. And I want to define these two very briefly, if I may. Life history traits are the key events in the life of an organism, of any organism. But here I have decided to depict the life history traits of a classical human lifespan. Individuals are born, they go to school. In certain countries, they get formally introduced to society at age 15 in the so-called fiesta de quinceañera. They can go and graduate after their education and go on to make a lot of money in the first highly paid job. This is where the audience, I'm sure, will be laughing, but we're missing that in the online interaction. If they choose to, they would then go on to couple up or live together or marry, you call it whatever you want, with another person from the same species. Then decisions come into play. When do you start to reproduce? How many reproductive events, aka babies, in our human species do you have? How much time lapses between each reproductive event? How much quality of investment do you put into each of your offspring? When do you stop to reproduce? Another very key important life history trait is the age at retirement in human societies. And apparently back in the 1700s, creepily enough, a really important trait was also the age at death, which still remains highly important because it inflects on everything that we do nowadays. But back in the day, with another extra layer of complexity, which is that it was a thing, it was in vogue to take photos of not just that event, but every single event in your life history. So the way in which I define a life history trait is any of the snap photos that prior to the advent of this guy here, smartphone technology, your grandparents perhaps would have gone on to take in order to be able to remember for posterity. What's important to keep in mind about life history traits is that they underpin, they determine, they give us a tangible record for what's possible. Here, for instance, you've got two species of rodents and orchids that take long periods of napping, dormancy, hibernation. So that's determined through life history traits. The mobility of certain birds and the long-term dispersal of certain plants is also determined through life history traits. The amount of reproductive investment that organisms put into their next generation, as depicted here in the two extreme cases, corals and orchids that can each produce 1 trillion propagules per capita per reproductive event. I'm sure that this audience will also be familiar with the record world Guinness holders of growth. On the top, you've got the blue whale, which puts on about 80 kilos. That's the average size, the average weight of your average European male per day. Or bamboos, which put on about 0.9 meters of height per day. So let that sink in for a second. I'm telling you that there are extremes across the tree of life that can sleep for a long time, can move for a long distance, can reproduce a lot, can grow extensively on a really short period of time, or as in the case of the Greenland shark and the bristlecone pines, live for a very long time. A paper that came out in Nature last year reported a conservative estimate of over 400 years for the Greenland shark. And some records indicate that the bristlecone pine, which is the Guinness World Record holder of non-clonal longevity, some of those individuals were alive and kicking by the time that the Egyptian empire was in baby steps. So we're talking more than 5,000 years. So it's remarkable to be able to use life history traits to understand what the perimeter of a three-dimensional space of life history strategies is. But it also, through trade-offs, that is limitations in how the limited energy can be allocated to reproduction, development, survival. It's also important to realize that these life history traits cannot just result into any strategy because of the compromise that I just talked about, the trade-offs. And because of that, because of limitations in terms of how the limited energy goes into either or a combination of these vital rates, the Darwinian demon has not evolved. And if it had, we would know about it because we would be completely taken over by whatever creature that is. Perhaps you could argue the closest thing that comes to a Darwinian demon is certain species of bacteria, or even better, us humans. But nonetheless, the biodiversity that we can observe in nature is the result of the lack of overlap between life history strategies. It's the niche partitioning of those strategies that is underpinned by life history traits. Also importantly is the realization that life history traits are universal, whether you're studying a species of spider in Japan, or the famous Greenland shark, or bristlecone pines, or lithops in South Africa. They all are underpinned by the same universal currencies of development, reproduction, and survival. And because they all are based on the same key demographic process, they allows us to carry out pretty sizable comparative analysis. Now, this is for the data, but of course, I couldn't talk about the data without talking about the theory as well. So the field of life history theory, which is a branch from evolutionary biology, has been devoted to understanding the drivers of variation in the life history traits and the emerging strategies that emerge when an organism invests on different key demographic processes. Information such as, is it better to just reproduce once or multiple times? Information or findings about how rates of migration and extinction shape biodiversity. Insights by Grind, for instance, regarding how species react to disturbances, or by Charnoff regarding how life history traits also scale with body mass, and many others, just here to mention a few, have been really fundamental in our understanding and our ability to predict which species may be the winners and the losers to climate change. And certainly my research capitalizes on that understanding. I apologize, obviously, it's a vast field and I cannot cite every single pertinent citation in that previous slides. But perhaps one of the most important tenets in life history theory is that of one ring to rule them all, as Lord of the Rings fans will understand. There's the fast low continuum, which was first posited by Steve Stearns, previous speaker from this series as well. The fast low continuum states that the life history strategies of any species can be predicted and organized along a single line, a continuum. At the end of one extreme, you've got species like copepods, for instance, that live very short, they grow relatively fast, and they reproduce young. And at the other end, you've got your famous species like bristlecone pines that grow really slow, attain high lifespans, and they don't reproduce much, although they do have a long reproductive window to do so. What's important to realize here let me see if this slide works. What's important to realize let me see if I can put this on. I apologize, pointer option. There you go. What's important to realize here is that a lot of this theory that goes back to 1950s, 1960s was only formally tested with the data that was available back then, decades ago. The data that was available back then, as you might have realized or guessed, was mostly that of humans, rest of mammals, and a lot of birds. But we didn't actually have access to the demographic data that would be necessary to characterize the strategies of many other species across the tree of life. And this has been one of my main tasks, and I would like to think one of my main contributions to science, understanding how strategies across the tree of life are orchestrated and how we can take this understanding to predict and forecast biodiversity into the future. Just so that you keep in mind, that line that we started with was in that little corner here in the red arrow. So we really know, or used to know, but a speck about the demographies across the tree of life. The word that Elizabeth highlighted before is very much encapsulated into this URL, so I won't spend much time talking about it. These are two open access databases that contain the demography of hundreds of plants and animal species respectively. They are contained in the shape of a matrix population model. If you're not sure what that is, I'm happy to explain that later in the chat, but just in a very simple summary, they encapsulate rates of survival, development, and reproduction, which is what we need to understand in order to describe the strategy of a given species. I also have another student co-advised with Tiffany Knight at IDIF, Sam Levin, who's also archiving two sister databases to Compadre and Comadre called Padrino and Madrina, which do the same thing for matrix population models, but for integral projection models. If you're interested in using IPMs for comparative purposes, I highly recommend that you touch bases with Sam and take a look at that URL. This is the URL for Compadre and Comadre. It's a fully open access data set. And hopefully in perusing that and surfing through it, what you will be able to realize is that it's pretty well spatially distributed around the globe. This is a world map highlighting the GPS coordinates of all of the studied populations across mostly animals and plants. We also have got species from other kingdoms, as you can see in different colors. And the usage of this information allows us to start to ask really global questions that, again, just like the fast law continuum, up until recently, we weren't really able to ask about the potential universality across the tree of life. The data, in case you're wondering, come from few people like me during my PhD. You can see a photo there prior to senescence, where I was collecting demographic data of a given plant species in Utah. People go to the field, they mark individuals, they obtain rates of survival, development, and reproduction, and they compile them into a mathematical object called a translation matrix. That information then makes it into Compadre and Comadre through an army of very brave students that I co-supervise. And what's interesting about these matrices is that through the help of a lot of really clever demographers such as Hal Caswell, Tulja Purkar, and many others, we can now decompose really meaningful, biologically meaningful properties such as mean life expectancy, the rate of senescence, the age at which individuals in a population start to reproduce, and many others at the population level, like the population growth rates or the rates of resilience. That is, if a population is perturbed by a disturbance, does it resist the disturbance? And if it doesn't, how quickly does it go back to equilibrium? So some collaborators of mine and I have been developing an R package, which you can see in that URL, called RH, or RAGE, because at times it was quite frustrating. And what this package does, it interfaces with any matrix population model to derive life history traits. These are the photographic memories that I was talking about before. And in this first line of research that I will present today, these life history traits are categorized according to different colors as per investment in population turnover, that's generation time. Investments in longevity or survival will be the same. The rate of senescence and the age at maturity. So that's how long do you live before you commit to reproduction. And then there's 4 different moments of reproduction that have to do with the intensity, the timing, the frequency, and the duration of reproduction. And last but not least, because this analysis was first done for plants, whether the individuals have the ability to grow and how fast, but also to shrink. And this is a key important factor to consider, particularly when studying organisms that, like plants, are modular. So many organisms that are modular and sessile can decrease in size from one year to the next. Corals do this. The entire plant kingdom, so long as it is long-lived, can do that as well. We then go on to put it in an n-dimensional space using standard phylogenetically controlled principal component analysis to ask whether the species is best explained by a single axis, as predicted by the fast law continuum, or whether we would need more than just one axis to explain the life, history, strategy, variation, in this case in the plant kingdom. What this approach does is it kills 2 birds, or 2 plants, really, if you allow me the pun, with the same stroke of the keyboards. It allows us to not only ask the question about the fast law continuum, but also to ask which forces, phylogenetic ancestry or environmental filtering, are most responsible for the patterns that you see. Here you've got 2 different plant species from the same genus Pinus or pine. They're really closely together within the phylogeny, obviously, since they are in the same genus, but actually environmentally, they are located in very different habitats. So the analysis that I'm going to show you now is going to allow us to ask the question, does phylogeny matter more than the environment, or vice versa, in determining what's possible life history strategy-wise for the plant kingdom? This is the PCA of the various life history traits that I described before. Here, I'm only showing PCA1 and PCA2 because using some sophisticated statistical analysis, it was determined that we actually needed two, not one axis, to determine a significant amount of variation. The first PCA corresponds to the fast low continuum. Each point here is one species. Species to the left tend to grow fast, whereas species to the right tend to live long and to escape from senescence. These are plant species, remember. So there's a trade-off between growing fast and living long. And that axis absorbs up to 34% of variation in the plant kingdom. The second axis has coordinated all main dimensions of reproduction in red, so the intensity, duration, the frequency, etc., of the reproductive events on the one hand, and it's trading off against the ability of the plant individual to decrease in size. The way in which we explain this is that in the plant kingdom, usually reproduction scales linearly with size. So if you decrease in size, you're not as reproductive. Together, this axis provides a first view into how rich life history strategies in the plant kingdom can really be. They explain up to 56% of the variation. And in the analysis that we run, the phylogenetic signal, which will tell you how important the ancestry is, was really low. And in posterior analysis that we have run, it's been determined that where the species lives is a much more important factor in determining the range of demographic performances and the range of this repertoire of strategies that can be available to any one plant. Importantly here too, we are able to predict key demographic properties such as how well the population as a whole is doing. So here we're depicting the position of every one species. This analysis, by the way, I don't think I mentioned before, there's 412 plant species. So 1 point is one species. And what we are doing here is we are color coding the population growth rate of that particular population according to whether it's growing in reds, staying constant through time in whites, or declining in blue. What you can see is that the second axis, the reproductive strategies axis, is a really good predictor of how well the population is doing. We can predict the long-term dynamics, but we can also predict how quickly a population will recover based on these two axes as well. So that's the rate of recovery, which is another handy property that we can derive from matrix population models. And one of my postdocs, Paul Cadevilla, just got published a paper in Tree a few days ago, which explains how these matrix can really be taken away to apply to conservation biology. So if you're interested in that, please go and check it out. From this analysis, what we can conclude is that species that have got the ability to decrease in size, or species that live fast, they are short-lived, have got a greater deal of resilience than those that do not have those two abilities. From the same framework, we can also predict and connect the demographic properties of these strategies to other disciplines, such as functional trait ecology. So SLA, which is one of the most widely used functional traits, can be predicted based on where the species is based on the fast low continuum. As you can see here, as you work from short-lived on the left-hand side to long-lived on the right-hand side, species tend to construct leaves that have got a lower score of SLA. Those are more expensive leaves to create, in other words. Here you have a depiction of what those two leaves look like. And by interfacing this framework also with the conservation status of the red list of the IUCN, we can also predict with an interaction term between the fast low continuum and the reproductive strategies continuum, whether the species will be critically endangered in red, endangered in orange, or going all the way to the opposite of demographic performance, that is invasive in blue on the top right. The analysis that I just presented was done for plants, but a postdoc of mine that I co-supervised in Trinity College Dublin performed a very similar analysis for Comadre, the animal database. And what he found was that, yet again, you need more than one axis of variation to explain close to 60 or 70% of the variation in these strategies of life histories in animals. And in case that you're wondering, we are there. So we humans tend to be relatively long-lived. We're definitely not the longest of them all. things like tortoises, for instance, live way longer than we do, and corals as well. And we are relatively semmelperous. Do not please that in this publication, reviewer number 2 got away with his or her demands, and we had to flip axis too. So on the top in this axis, you've got semelparity, species that reproduce just once or very few times. And at the bottom, you've got highly iteroperous species with a long reproductive window. But we, of course, didn't stop there. We have got a really poor understanding of the demographies and the life histories of aquatic species. You could argue that this has to do with convenience, and I would agree with that. But one thing is true in that due to climate change and ever-growing human populations, we really ought to understand how well the stocks of the ecosystem services that lie in aquatic ecosystems are going to be doing now and into the future. So understanding whether the demographies are viable or not is of utmost importance. And this is something that one of my postdocs, Paul Capdevila, recently did, comparing the life history strategies of aquatic, that's both marine and freshwater, versus terrestrial species. What we found there is that you also again need 2 axes of variation. So the fast law continuum doesn't explain as much as one would have predicted in classical life history theory, but also what we found was that for the sample, and I do want to highlight the word there, sample species that we have access to from aquatic ecosystems, those species are displaced towards the left. So they are shorter lived relative to other species. This might very well be the case, or this might be due to the difficulties, the challenges of going very deep into the sea to sample abysmal areas. But stay tuned to this information. Shorter-lived species in aquatic ecosystems. This is going to be really important for ecological forecasts in just a second. In 2017, I redid some of these analysis with plants. This analysis contains 615 plant species. And in doing so, the patterns became even more robust. You've got 2 axes, the fast-low continuum and the reproductive strategies continuum. But I also realized that there was almost like 2 clear-cut diagonals running on the second quadrant and on the third quadrant of the PCA. And that got me to think, why so? Are combinations of life history traits not viable in those 2 spaces? Or are we simply so biased in terms of what we understand and what we have stored in Compatriot and Comadre that we don't have information for that? So the current working hypothesis is that on the bottom left, if you don't live long and if you don't reproduce much, you're pretty much predestined to extinction. So a strategy that is both short-lived and not very reproductive makes no sense. However, you could potentially predict the viability of some elements, as a chemist will tell you, using the life history periodic life table on the bottom right of this PC axis. So what I'm predicting is that there are indeed species that are very long-lived, that are not very reproductive and have got the ability to decrease in size. And these are species for which, for whatever the reason, we have not sampled yet. And this is what I'm using actually as a working excuse to go to some really exotic places, such as South Africa and other places like Australia, to sample animals and plant species. Together, nonetheless, these 2 axes, the fast-low and the reproductive strategies, which has got extreme iteroperous at the top and extreme semelperous like salmons at the bottom, provide a new framework. And they provide a new framework that is, as I mentioned before, very analogous to the periodic table of the elements. To the left, how long-lived. In chemistry will be how many levels of electrons you have. And to the bottom, the degree or the window of reproduction, which will be analogous to just about how many levels of electrons you've got in the atoms. Can we use this framework to predict? And can we actually find new species like that technetium elements that I mentioned before that have not yet been sampled demographically? Well, tennessine was only discovered about 11 years ago. And this is coming from a discipline, chemistry, that is far older and much more established than ecology. So my claim is that ecologists probably would do well in studying things in addition to lions and dandelions, sure, things that don't look very much like us and branching out into the rest of the tree of life. Now, moving on to the climatic forecast part of this presentation. One of the most pressing needs for ecologists and for societies is to be able to understand how our natural capital and the ecosystem services that flow through the natural capital will be affected or not by different scenarios of the IPCC. And I think that I don't really have to convince anybody in this audience that climate change is real, climate change is tangible, and climate change has done an awful list of very long things to biodiversity, not always positive. In research led by one of my previous PhD students, Maria Panev, we did a literature review for all mammal species, examining the effects of climate onto their vital rates or demographic rates. Those are survival, growth, and reproduction. We identified 106 such studies corresponding to 86 mammal species. And what you can see here is that the studies, the GPS location where they took place, are not necessarily located on the areas that are predicted to be most vulnerable to climate change. So the choices that authors are making to study different mammal species around the globe, mammals being one of the best demographically studied group in the tree of life, is not very well coupled with where we need those studies to take place. That's the first take-home message. Now I'm going to present you what's actually a potentially rather confusing figure. It's a really rich figure, so I'm going to walk you through it step by step. Here you can see, out of this literature review, the number of studies that looked at demographic rate times climate interactions. The darker, the more studies that looked at the evidence. I'm going to show you quite a few mammals in a second, but here we've got a meerkat. Meerkat that is one of the study species that Maria has studied in her postdoc recently. And what we found in this meta-analysis is that most of the demographic information connecting the vital rates of survival, reproduction, and growth to the climate were non-linear. So really difficult to predict, in other words. If we expand this to other mammal species, and here you've got a repertoire out of the dozens of species that we looked at, there's not a clear pattern that emerges from this. There's a lot of interactions in terms of how the climate can predict positive or non-linear or neutral or negative, as explained on the left axis, but there's not a single way in which climate is actually affecting. So what this says is that just looking at the vital rates of any one species is not really a useful approach to do comparative demographic forecasting. When exercises like the one that I'm going to present you in a second have been performed in the past, authors have assumed that, yes, the climate is going to become more extreme, but that the transitions between different climate events is going to take place at random or stochastically. That is, the environments can switch back and forth between a year of good conditions and bad conditions in an IID, or independent and identically distributed manner. But one of the key findings from the IPCC scenarios is that the climate, which is already correlated, is expected to become even more so positively autocorrelated. What this means is that if you happen to be studying a population that is starting in a good environmental year, congratulations, you will most likely be seeing that condition for a long time. However, if you study in a population in a bad environmental year, you're likely to be sampling that for quite a few times before the population switches over to a good year. And the predictions and the theory from life history that we have to do ecological forecast has simply not developed enough to take into account the positive autocorrelation. So what Maria and other collaborators did is we looked at the life history traits of 454 animal and plant species. We derived their life history traits, intensity, duration, frequency of reproduction, and survival and generation time. We put them in a PCA, and here you can see that the first one explains 34% of the variation, whereas the second one explains about 33%. So these two axes together explain a lot of what the strategies available to plants and animals are. What I'm going to show you next is how sensitive the long-term stochastic population growth rates of each population is to an increase in the positive autocorrelation of the environment. Now, you do realize that these are matrices that come from real population, but the simulation is not really actually linked to the environment, which is a thing that I'll show in the next piece of research. Here, we're just doing simulations classifying years as good or bad, and we're increasing the frequency of good to good and bad to bad. What you can see here, a repertoire of both animals and plants that made it into this study. The bigger points are the points that are more sensitive to an increase in the positive autocorrelation. That is, they're more vulnerable. These are populations that are more vulnerable in what the IPCC predicts, forecasts it's going to happen. And these are short-lived species. Remember what I mentioned before about marine species being shorter-lived than terrestrial species? Where we are in trouble if that finding is true, if that finding is not biased by sample size availability. In a paper currently reviewed by another postdoc, Aldo Compagnoni, that I co-supervised in IDIV, we evaluated from 165 populations coming from 62 plant species worldwide, for which we had demographic data for at least 7 years. We examined how the climatic extremes might shape the population growth rates. So what you have here is on the x-axis, water availability index for various populations on the top panel, and the mean annual temperature of those populations for the bottom panel. And on the y-axis, you've got the effect of precipitation or temperature anomalies onto the population growth rate on a log scale. Population growth rates that are positive indicate the population benefits from that disturbance, from that climatic anomaly. Population growth rates that are negative indicate that the population takes a hit in a negative way. What you can see is that these plant populations are massively being affected positively if they lived in arid lands, but they're being affected negligibly regardless of where they live by a decrease or an increase in temperature anomalies. Can we use then life history traits to predict the responses? Well, the answer is yes. What we find here is the absolute effect of precipitation onto the stochastic population growth rates, y-axis, can be effectively predicted by how long-lived and the generation time of that species. Species that have got a shorter generation time benefit more from the anomalies. We also use the degree of parity, which is the index that we use to classify species according to extremely semialberous or extremely alteroberous or anywhere in between, as a response variable to these climatic anomalies. And what we found is that there was a borderline significance, but it wasn't nearly as significant as generation time was. So taken together, what this means is that out of the sample life history trait space, which kind of has the shape of a two-dimensional ice-cream cone, we might be entering a new phase in the Anthropocene where climate change might be biting from one side of that ice cream cone. And the side that is biting away from is that of short-lived species. Just very briefly, in the interest of time, I'll mention in passing that we currently have evidence of there being a third axis of variation that has to do with whether the species undergoes senescence for reproduction or not. There's a paper there that we have deposited recently in my archive that can give more information. So pretty much a species can undergo senescence for mortality, but no senescence for reproduction or vice versa. And together, this information can provide us with an even better understanding of what this so far 2D, soon 3-dimensional framework of life history trait strategies can help us predict. We've provided a pretty substantial list of evidence that understanding how long-lived and how often species reproduce can help us predict anatomic traits, demographic performance, the evolution of senescence, how endangered species can be, etc., etc. So I would argue that this life history trait framework, which again is heavily inspired by the periodic life table of elements in chemistry, can provide a really insightful amount of discoveries for ecology and evolution. That environmental filtering is way more important in what species can do life history traits and life history strategy-wise, and that we can, for the first time, use demographic data at a massive comparative framework to implement IPCC projection forecasting. I would like to end by thanking my various funding agencies. Very importantly, everybody in a working group called S Apropos, which I lead at the Center for Biodiversity in Germany, IDIF. And also, if there's anybody interested in this type of research who are currently looking for PhD opportunities, summer internships, or postdocs, please do get in contact with me, particularly if you come from a diverse background. Thank you very much.
Fantastic. Thank you very much, Rob, for a really interesting and thought-provoking talk. There's a lot of questions which are provoked from your research, for sure. While the questions are starting to come in from our audience, I thought I would start off with one for myself. So it's quite curious when you show for the plant data, that plants that were on the live fast side of the fast slow continuum seem to be more resilient. And I was just wondering if you might be able to speculate why this could be the case, and why the fast strategy could be beneficial in that scenario.
Yeah. So I think that when asking resilience, what makes a species more resilient, is very important to also follow the question with resilient to what? The way in which resilience has been measured in that analysis was resilience to the return to demographic stability, okay, which might be different from the resilience to return to positive population growth rate. And actually, the machinery that would allow us to derive all of those metrics, we're currently working on those. But in this specific analysis, the question that we ask is, how quickly does a population go back to stationary equilibrium? And what we found out in that analysis is that there's pretty much two ways. Either you pull from propagule or you pull from yourself. Allow me to elaborate further. What I mean by that is that if you take a hit by a perturbance, a disturbance, say a fire, there's typically different strategies to a fire. You've got the seeders and you've got the resprouters, right? So the seeders are the species that don't tend to allocate a lot into their own longevity. They allocate more into reproduction. Whereas the resprouters are the species that typically have got a storage unit below ground. So they're more committed to their long-lived style. They don't elaborate or invest so much into reproduction. So what that analysis does is it confirms that responses to fires, as well as responses to many other disturbances alike, have got two different non-exclusive strategies that can give you the same benefits. Either live short, die young, and invest into the next generation, or in the second case, decrease in size, as you would if you get burnt, but you survive below ground, and then regrow from that still alive individual stock.
See, that's really interesting. So obviously the links between the reproduction and the growth will feed into that for sure. Great. So I think we'll move on to some questions from the audience now. Mm-hmm. So the first question asks, can life history theory predict how many species there are? Or conversely, are we unable to say how life history space is filled?
I think it could. I don't think it's been asked up until recently. We certainly are really interested in those type of questions. I think that two things are required in order to be able to address this very global and important and timely question. The first one is to understand whether and how many different life history strategies can coexist. Because if the niche space that has to be distributed across different species is very narrow, then that wouldn't give rise to as many strategies to coexist as others, right? The second one is simulation. So you can go outside to the world and commit yourself to 10 years of demographic sampling, which I would argue is valuable, but really painful. I've done that myself, okay? Or you can just build on the vast amount of demographic information that we have, use that to create virtual species, create demographic models that underpin the trade-offs that have resulted into those species, and then explore how trade-offs between survival, growth, and reproduction can shift around to produce a population that will have a population growth rate greater or equal to 1. That is, in fact, something that one of my co-advised PhD students, Simon Rolfe, is currently looking into. And we're very excited to be able to take that approach further to look at how that hyperdimensional volume is going to be perhaps shrunken? And if so, in what way? By different kinds of perturbances.
Thank you. That's really interesting. And following on from that question, we have another question from the audience, which asks, to what extent do sources of variation, genetic, environmental, help predict population response to change, such as persistence and functioning? And can they be used in the framework as well? For example, some species might be rescued by evolution.
So one of the things that we haven't yet worked out, or we haven't published, that is how to include the evolutionary angle into this framework. Work being done in my department actually by Tim Coulson, but also by other folks around the globe, has shown very promising approaches to incorporate ecodynamics into a demographic framework for one species at a time. Now, in order to be able to do that for as many species as we currently have data for, we need to really understand the genetics and the inheritance of those species and the modes of reproduction as well. That's something that we're currently working on. We're constructing an ancillary database to Compadre and Comadre called STRAIGHTS, whose main objective is to acquire information that will help us to address global questions like this one.
Fascinating. It would be interesting to see the interplay between evolution and ecology on the multi-species level, for sure. That's really interesting. So we have a question which asks, it says, short-lived species are in trouble from climate change, demographically speaking, but they also disperse further, as shown by Beckman et al. 2018, and can also evolve more rapidly. So is this how they may deal with climate change?
It's tricky to answer that question because how far you can disperse is also determined by the environment in which you live. So you can, all things being equal, you can imagine two different species that disperse far or not so far, but they are stuck in a valley of a mountain, right? So the elevation gradient that they have to overcome will also constrain what's available to them. And so I think that in principle, I could make some predictions without taking into account the terrain ruggedness. But obviously that would be like throwing the baby in the tap water. I think that the inclusion of the 3D-ness of the environment where the species lives is probably as important, if not more, than what the species can do.
That's interesting. It's almost like that would be included within the environmental aspect of things, whether they can disperse like within their immediate environment or even further afield, I guess.
Yeah. So dispersal kernels is something that folks like James Bullock have been acquiring data for a pretty long time. James was one of my co-authors in that paper that was mentioned before. And we're also very interested in taking into account how the dispersal can be affected or constrained by the shape, concave or convex, of the location where the species has been studied. But of course, we're talking dispersal. So we're talking about plants, I guess, right? Or fungi. But the same question could be also applied to animals, mobile or not. So really, I think that the question is really important, and it really requires that we assess even more globally, not just plants, but perhaps also animals.
Yeah, definitely. And actually, following on from that, we have a question which asks, so why do you think there is a link between development and lifespan senescence, which seems to be pervasive across plants and animals? And could this be indication of some sort of common cross-taxonomic mechanism?
Metabolism, I think, is the key. I think that metabolism is at the same time underpinned by available resources. And this is something that is often not very much looked into from a comparative perspective. There's this theory that goes back to Aristotle, as you probably know, that states, if you want to live long, don't eat too much, in simple words, right? But hold on a tiny second, I apologize. Okay. Sorry, my computer's giving me some headaches because it's at 7% battery. I think I should be fine though. Am I just connected to? I apologize. Am I just connected to the charger? That's right. Can you see me? Okay.
Yes. Yeah, that's fine. No worries.
Okay, fantastic. So metabolism underpins everything that we do and that we can do. And there's a trade-off, as I mentioned before, between development and longevity. You can grow fast or you can develop fast if resources are plenty. Right? all other constraints being taken care of. When I mentioned the World Record Guinness of longevity, I mentioned species that live in really nutrient-poor environments. So I mentioned the Greenland shark. Greenland waters are really nutrient deficient, and bristlecone pines that live in drylands, also really nutrient deficient. These two species grow really slow, extremely slow. And I mentioned before that there was a trade-off between growth and development. So I argue that the ability to, one of the possible mechanisms anyways, to escape from senescence or to postpone senescence is to be constrained nutritionally by the environment where you're living.
Yes, and then I guess that would make sense with the dietary restriction, caloric restriction studies as well on a smaller scale. That's really interesting. So you mentioned in your talk actually about this third demographic axis of reproductive senescence. I wonder if you could expand a little bit more on that. And the question asks, is there reproductive senescence in plants? And what could be the trade-off there, i.e. the benefit of living long without reproducing, for example?
So on that note, since you're asking for it, let me perhaps share about this screen. Can you confirm with me whether you can see that?
Yes.
Okay, fantastic. So this is one of the figures from a publication led by one of my PhD students, Mark Roper. This is just a simple snippet of 8 species, both of plants and animals. The species to the left undergo senescence. They are marked in blue in the silhouettes. The species to the right escape from senescence. That little H letter, that little H index, is key fits its entropy. That tells us whether LX, which is the survivorship, is declining mostly at the end of life, which is what you would predict if a species undergoes actuarial senescence or not. So if you are more likely to die at the end of your life than at the beginning in a whole cohort study, then you're told to be undergoing actuarial senescence. The assumption in many comparative studies has there been that if you undergo senescence for one fitness component, that being survival, you would also undergo senescence for the other one, which is reproduction. And what we show in this paper is that, also using data from Compatra and Comadre, there's absolutely no correlation. The shapes of survivalship here in reds, so that's the LX, and per capita reproduction, which is the blacks, are all over the shop. In the next slide, perhaps I can even depict that in a more drastic way. I do realize that I'm breaking rule number 1 of our presentation, which is not show more than 1 or 2 panels. But the point of this presentation here is to really show you just about how diverse the survivalships and the reproductive rates can be across the tree of animals and plants. How that happens, we've got some ideas, but we don't have anything that is robust enough. So allow me to speculate. We are hypothesizing that the degree of indeterminism is key to set these differences. So as you know, there are some organisms that upon birth, they've got a given number of bullets determined for reproduction, right? You and me among them. But there's many organisms of reptiles and some most plants, in fact, where the reproductive agendas are indeterminate and they can be triggered by the environment. We suspect that that might be a really good predictor for the decoupling of actuarial senescence from reproductive senescence.
Ah, I see. that's really interesting. Oh, wow. Yeah. So, yeah, it's definitely not the case to assume that all traits senesce at the same rate or even senesce at all, I guess. And so it's important to take the reproductive senescence into account.
Indeed. I mean, I've got, if we were not going through the pandemic, I would be probably not speaking with you and your audience today, but I would be probably collecting field demographic data of Mediterranean shrub species in Barcelona right now, first week of July. That's when I will be doing this field work. I've been collecting demographic data for 600 individuals of this plant species, Cistus albidus there. And so I have developed a pretty good idea of how different demographic processes change as organisms age. Even for the individuals that weren't born into the population, as I wonder, individuals that were already existing in that population, I can already guess how old they are based on some morphological and dendroecological proxies. So I know pretty much how old every individual is in that population. We've been following survival as we have been following growth rates and shrinkage rates. We've been following probability of reproduction, number of flowers, number of seeds, number of capsules, number of seeds. We've also evaluated the germinability and the viability of every single seed within every single individual, multiply 600 individuals, so a lot of work there. And what we have found actually is that the species undergo senescence for some vital rates, but not for others, even within reproduction. So the species becomes more likely to reproduce with age, but the quality of the seeds that it produces goes down with age as well.
Okay, that's really interesting. So it could be some trade-off between viability and actual number produced, potentially some sort of quality quantity trade-off, perhaps.
I suspect so. Great.
That's fascinating. Thank you. So I think we've got time for another question from our audience. There's a question which asks, it seems as though shorter-lived species are struggling demographically due to climate change, but is there much evidence to suggest that climate change could have positive effects on other species, e.g. at the other end of the fast-slow continuum, for example?
So yes, there are some sweet cases where anomalies can actually be good for different species. I spent about 10 years of my short academic career collecting demographic data on a plant species in the Great Basin Desert in Utah. And what collecting 10 years of data at a given location does for you is it exposes you to everything that can happen in that field site in terms of climates. So I've been rained on, I've been snowed on, I've had some really hot days, and so have the plant species that live there. I collected the demographic data to parameterize a demographic model to do ecological forecasting. In that specific region, the climatic models predict an increase in precipitation rather than a decrease, as they do in most desert ecosystems. Again, this is the Great Basin Desert, so it is a desert. It's a cold desert, though. So, and this increase in the precipitation has to do with the disruption of El Niño cyclicity. So with the disruption, more rain monsoons are going to make it into the west of the US and percolate into Utah. The increase in precipitation is also associated with a greater rate of flowering and germination in this plant species. So in the case of that one specific plant species, climate change predictions are good news.
Oh, wow. That's interesting. It's fascinating. It'd be cool to hear more about that at some time. But yeah, thank you. So I think that's all we have time for today. But it's a great pleasure to thank Rob once again for a truly fascinating talk. Thank you, Rob.
Thank you so much for having me.
Thanks. So before we finish, I'll just remind everyone that our next talk will take place on Wednesday this week. And will be presented by Professor Christopher Wheat on the evolution of life history strategies in butterflies. So we hope that you can join us then, same time and same place. And in the meantime, check up on our updates on Slack and our Twitter feeds and spread the words about our seminars. Thanks for taking part, and until next time, stay safe and see you again soon. Goodbye.